So here is a cell with two pairs of chromosomes. There's a red pair and there's a yellow pair. And the idea is that I've actually coded them so that you can tell the difference between each one. Because what would have happened is that you would have gotten each of your chromosomes, you would have gotten one from one parent and one from the other. So this is the cell in interphase before anything exciting has happened. Then the cell gets an option to go into the DNA synthesis phase of interphase called the S phase. And in that moment, what will happen is that the cell is going to go ahead and make exact copies of the chromosomes it has. So what I'm doing now is pretending we're in S phase and adding the rest of the DNA that would be made in our copy in here so that we can see what's going on. Okay, but right now it still looks kind of like a mess. The reason why it looks like a mess like that is because we're still in interphase. Then we get into prophase. In prophase of mitosis, what's going to happen is that the nuclear membrane disintegrates, the chromosomes condense, and at that moment, you're actually able to see them. So using these as an example makes it a little bit not exactly accurate, but we could see them. So voila, I can see no nucleus and I see the chromosomes with their duplicated pieces. So these are sister chromatids, they're exact. These are homologs where these are similar, but the original one of both of these would have come from one parent and these came from the other parent. One, and then we made a copy. So now we're in prophase. If we're in mitosis, then we line up in metaphase. So we'll go ahead and start lining everybody up. Okay, and it doesn't really matter, but the idea would be they're all lined up in a line, which is bigger than what we have here. Okay, all lined up in the middle so that each place the sister chromatids could be separated. So when they're lined up in the middle, it's metaphase. Then in anaphase, we pull them apart from each other. Okay, and this would happen all at once, but my hands can't quite do it. So I'm pulling my copies apart from each other. Okay, and you can see them all pulling. You can see them kind of getting pulled from the middle at the centromere part. Okay, that's anaphase. When we get to telophase, what we're doing is these guys are going to make a new nucleus. And this one over here would make a new nucleus. And what you're going to notice is that we're making those extra pieces. It's kind of like what we started with, only two of them now. And then in cytokinesis, I pinch off. This is one cell with its nucleus. Here's the other one with its nucleus. And then that's going to be mitosis. And if you look real close, we went ahead and got copies of everything exactly perfectly, which is the purpose of mitosis.